I've colored it in yellow for this diagram. Dad's copy has a lot of, I'll show you the genes in a second, but UBE3A, one gene for instance, is silent on dad's copy, but it's expressed on mom's copy. So individuals with Angelman syndrome are missing mom's copy, and they're missing UBE3A, as well as other genes. Individuals with Prader-Willi syndrome are missing dad's copy, and several paternally expressed genes there. But individuals with DUP15Q syndrome can have one of a variety of different manifestations of the duplications. So one is maternal interstitial duplication, and this means that there are two copies on mom's chromosome 15 of the imprinted region that are in tandem. Maternal interstitial triplications then have 3 copies in tandem on mom's chromosome. And then the most common is the isodicentric chromosome 15, or IDIC 15, where there's an extra supernumerary chromosome that contains 2 copies of the imprinted region in between 2 centromeres. And finally, there are paternal interstitial duplications, but this is really a different disorder. I've stepped back from saying that they're unaffected because as I understand, there are some phenotypic presentation in that population as well. But importantly, they have the same duplicated region, except it's paternal instead of maternal. And you'll hear a lot of the speakers going forward talk about UBE3A, because thinking about the maternal interstitial duplications, the real difference between the maternal interstitial duplications and the paternal interstitial duplications is the extra expression of UBE3A. That's the only imprinted and maternally expressed gene in the locus. And I'll say this again in a different way. So the reason, these are the genes that are in the imprinted region, as well as those that are flanking. The reason that these duplications and deletions happen is because there are breakpoints, shown here as BP1, BP2, so on and so forth. And those breakpoints are places where the DNA is repeated and the chromosomes misalign during meiosis. And due to that misalignment, breakpoint 1, for instance, can fuse with breakpoint 3 and mediate a deletion of that region. But it can also break and cause a duplication, and this is why we're here. So the reason that the parent of origin matters is because several genes in this region are expressed from dad's copy. Those are shown in blue. And one gene, UBE3A really, is expressed from mom's copy. There's another gene, ATP10A, that's imprinted, expressed from mom's copy in some individuals. But to be honest, that's really poorly understood at this point. Like I said earlier, I should say that the parent of origin specific expression is set up by an imprinting center, and that's here at the SNRF-SNRP-N gene. It's a differentially DNA methylated region that's unmethylated on dad's copy, methylated on mom's copy, and that's that differential methylation really drives the pattern of gene expression for this region. So then the other thing that I need to tell you is that there are several non-imprinted genes. Non-imprinted means they're expressed from both parental copies that are adjacent to this region. They're also deleted or duplicated in the various syndromes. These include a cluster of GABA receptor subunits, HERK2, which is another ubiquitin ligase like UBE3A, as well as some other genes that are present in some individuals with DUP15Q syndrome, but not, that are duplicated in some individuals with DUP15Q syndrome, but not all. So I also want to point out that there are the duplications that we're talking about here, those that involve breakpoint 1, 2, and breakpoint 3, 4, 5, cause DUP15Q syndrome. And the deletions that can involve the same breakpoints that cause Angelman or Prader-Willi syndrome, these are all fully penetrant disorders, meaning that everybody who has the duplication or deletion are affected with the disorder, okay? And that's kind of an important point, because there are individuals that have duplications and deletions of breakpoint 1, 2, or breakpoint 3, 4, 5 that involve those, and those confer perhaps increased risk for neuropsychiatric phenotypes, but they're not fully penetrant. And so they're different and not necessarily the same disorder as we're talking about today. And importantly for what I'm going to tell you about going forward, My lab uses iPS cells to study these disorders. Knowing that they're fully penetrant is hugely helpful because I know that if we make iPS cells from a patient with these duplications or deletions, we know that they're affected with a disorder. They have the characters that are required for them to be affected with a disorder. Whereas we wouldn't necessarily know that if we tried some of these other duplications or deletions. So my lab, like I said, uses iPS cells to study these imprinting disorders. And what we do is we take various somatic cell samples, fibroblasts from skin punch biopsy, cord bloods, peripheral bloods. We've done it from urine. We've done it from dental pulp stem cells. We can reprogram them using a variety of different methods, but overall you express stem cell genes in a non-stem cell, and eventually the cell becomes a stem cell. And these stem cells are functionally equivalent to human embryonic stem cells in that they can become any tissue type in the human body, theoretically. We're most interested in neuronal differentiation because we're studying neurodevelopmental disorders. So we use a couple of different differentiation protocols that I'll get into in more detail in a second. And we get these beautiful neurons in a dish. We can't do behaviors. We can only look at neurons and ask about neuronal function. And Eric Levine will tell you a little bit more about neuronal function later. But our iPS cells, importantly, we're not using these for replacement therapy. A lot of times you hear about stem cells and you think that we want to replace the cells that are missing. There aren't cells missing, to our knowledge, in the brains of kids with Dup15q syndrome. But rather, we're using them for disease modeling and for drug discovery to understand what's going on in Dup15q and to help figure out what are the genetic contributions to Dup15q so that we can find targeted therapeutics for the disorder. So one of the other important things that I often gloss over, and I'm reminded by NIH reviewers and whatnot to not gloss over, is the characterization of our iPS cell lines. Good researchers will be very careful with the characterization of their iPS cell lines because we can't take for granted that they're always intact and as we think they are. And so what my lab ends up doing is once we've derived iPS cells and every 10 to 20 passages thereafter, we will go through this cadre of quality control steps. We stain for pluripotency markers to make sure they're still stem cells. We do DNA fish or karyotyping to confirm the duplication. In this case, you can see here these are the chromosomes with that little IDIC 15 chromosome shown here. Karyotype also to look for genomic integrity to make sure there aren't any other duplications, deletions, or translocations. And then we've more frequently been doing SNP arrays to double check the genome integrity, but we also like to do this to make sure that the duplication breakpoints are the breakpoints that the patient has told us they were in the first place and to make sure that that hasn't changed. So lucky for us, the imprinting center is the other place we have to look carefully for quality control. And the correct imprinting center at this differentially methylated region at SNRF-SNRP-N is really exquisitely maintained when we reprogram somatic cells to iPS cells. And we've looked at several Angelman cell lines and Prader-Willi cell lines and now several Dup15q cell lines, and almost always it's maintained well. And it doesn't seem to change over passage in culture and differentiation. So we're fortunate in that. And what we can do is we do quantitative DNA methylation assays to look at the amount of DNA methylation. And so for instance, in a maternal interstitial duplication, you have 2 maternal alleles and 1 paternal allele, so 2 methylated dots and 1 unmethylated dot, and altogether that gives you 66% methylation because you have 2 that are 100% methylated and 1 that's 0% methylated, and that's why you're 66% methylated. And we see this with our maternal interstitial duplication line. Our paternal interstitial duplication line comes in at about 25% methylation, which is a little bit lower than the expected, but that is what it is. And then our IDIC-15 lines all come in about 75% methylation. And thus far, even when in the case of Prader-Willi syndrome, we've wanted to change that DNA methylation, we haven't been able to. So I think that this methylation imprint is actually quite sturdy in culture. I mentioned earlier that we differentiate. We use a variety, we differentiate our iPS cells into neurons. We do a variety of different neuronal differentiation strategies. One is embryoid body-based differentiation. This one's developmentally correct, but can have non-neuronal cell types that can influence their function. So we don't use this a whole lot anymore, although some of our data previously has been generated using this strategy. My lab's favorite is this dual SMAD inhibition monolayer differentiation because it excludes other non-neuronal lineages, but this differentiation strategy is very scalable. And it includes glia, which are important for maintaining or establishing the proper neuronal function in the first place. And then it's sexy but somewhat useless for us to do these cerebral organoids. These are self-organizing really mini brains in a dish. They're self-organizing and they have great neuronal connectivity, but they're highly heterogeneous. And so for me, who studies genetics, the heterogeneity makes them difficult to use. And I think, I think these really sexy technologies, we have to push researchers to use them appropriately and not just use them because they're a sexy technology. So, what my lab really does is we've been trying to use the iPS cells to understand the disorder and for drug discovery. And so one of the questions we've tried to ask is, can we use gene expression as a phenotype? Because mouse models are fantastic in that you can look at behaviors, right? Cells in a dish don't really have behaviors per se, although Eric will convince you otherwise when he gives his talk. And so we've tried to ask, can you read out gene expression as a phenotype? Because, and it's also very quantitative. It's unbiased and quantitative. We can look at the genes that are expressed in a cell culture and see whether, and count whether they're different. So in the past, we've published on this in Molecular Autism back in 2014. We've done RT-qPCR and Do15Q neurons to look at the genes in the imprinted region. We've also looked at, as well as the adjacent genes. And we've also performed, like, at the end of the day, probably a pretty crappy RNA-seq to compare Do15Q and Angelman to figure out, like, do they have a transcriptome phenotype? And so we want to look for these phenotypes because we also have looked at ways of modulating gene expression using both small molecules and antisense oligonucleotides. Small molecules like mithramycin can change the expression of genes like UBE3A, and this is a dose response curve for UBE3A expression after neurons with Do15Q syndrome have been treated with the drug. It reduces UBE3A in a dose-dependent manner back to the level of typical developing neurons. But mithramycin has a lot of off-target effects. Some of those are good. It also happens to reduce GABA beta 3, and that was just one that we happened to look at. I do keep thinking that we should come back and revisit mithramycin to see if it would be more useful than we've assumed. But again, being geneticists, I'm happier with targeted approaches like antisense oligonucleotides. We've identified ASOs that work well to reduce UBE3A in a dose-dependent manner, HERK2, GABA beta 3. We thus far have not developed an ASO that works well against GABA alpha 5, but in some mechanistic studies, we understand better how ASOs work. And so we may be able to revisit this and find one that works for that particular gene. But what's cool about these is they also work in a dose-dependent manner. And we can achieve an intermediate knockdown of UBE3A or the other genes where we're not totally getting rid of all of it, but getting rid of enough to approximate what we think a typical level is. And so all of these gene expressions are relative to a typically developing neuron. And I should have said up front that these are all IDIC-15 treated neurons, stem cell-derived neurons with these antisense oligos. So we have potential therapeutic approaches and things that we can study. And Eric's going to tell you, I think, a little bit more about phenotypic rescue, real phenotypic rescue. But again, I'm stuck on this idea of can we use a transcriptome as a phenotype, since I don't know how to do physiology, I know how to measure gene expression. So I'm going to walk you through some lessons we've learned from Angelman syndrome in this realm. And so my lab, Angelman syndrome is an easier disorder to study because it's a single gene disorder. And so what we've done is we've taken iPS cells from individuals with Angelman syndrome. In this case, this family has 2 boys who are affected with Angelman syndrome due to a missense mutation, meaning that it just changes the protein sequence, but the protein is there and it's expressed at normal levels. They have a missense mutation in UBE3A. It's really a 1 nucleotide change. It's from a TTT to a TCT. We've used CRISPR-Cas9 to change that C back to a T and correct the genetic defect in these the cells from one of these boys. In parallel, we've deleted mom's copy of UBE3A and human embryonic stem cells to make another copy that more closely approximates the mouse model, but also just gives us an independent pair of stem cells, and these are isogenic, meaning the same background genetics, Angelman and Angelman cell lines, Angelman and typically developing stem cell lines. When we look at the electrophysiology that we've done in collaboration with Eric Levine's lab, we can see that they have the known phenotypes seen in Angelman neurons. They have a more depolarized resting membrane potential. That seems to be the primary phenotype. They have delayed maturation of action potential firing, and alterations in spontaneous excitatory activity. And I should mention, since he's here, James, this is work that was really pioneered by James Fink in Eric's lab. So we know that we can correct this, we can use CRISPR-Cas9 to correct the cells, we have isogenic pairs, and we can use them to see known phenotypes. And so these were a perfect platform to ask, can we use gene expression as a phenotype? So what we did was we generated neurons from these isogenic pairs. The Angelman ones are here clustered together, and the corrected and typical human ES cells are shown, I guess, on your right. And we differentiated these into neurons and did RNA-seq and quantified the gene expression in these. And we did some pretty extensive informatic tricks. We used surrogate variable analysis to get rid of background, to get rid of changes that are due to batch effects or various things that we can't put our fingers on, know what they are a priori. And we asked, what are the genes that are differentially expressed when you consider this whole cohort? And ultimately we found 855 differentially expressed genes, 507 upregulated and 348 downregulated in Angelman syndrome. And that's for a single gene disorder. And so when we looked a little bit deeper, the other thing we learned is that these differentially expressed genes are not direct transcriptional targets of UBE3A. UBE3A is a ubiquitin ligase. It marks proteins for degradation by the proteasome. And so this is a scatter plot from our RNA-seq that I just told you about, and you can see all these red dots are the differentially expressed genes. The one thing that stands out from this is that, first of all, they're not high magnitude. They're not a high magnitude change. This is the log fold change here on the y-axis. They're actually subtle changes, but they're significant. On the other hand, we also did RNA-seq, in which we took neurotypical 12-week neurons and we treated them with an antisense oligo to knock down UBE3A, and we knocked it down lower than 80%, so it's a substantial knockdown. When we did the RNA-seq in those 3 days following the antisense oligo treatment, there's one differentially expressed gene, and that's UBE3A. So we don't think that there are direct transcriptional targets of UBE3A for this reason. But really these more subtle changes that are due to neurons developing with and without Angelman syndrome. And so whatever goes wrong in the first place, the neurons are developing and compensating to that defect, and that's the resulting quote-unquote phenotype that you'll get. This is like one other piece of data that suggests that UBE3A does not directly bind chromatin. I seem to be the queen of negative data, but we did a lot of different ChIP-seq using 2 different antibodies and several different cell types, neural precursor cells, stem cells, and neurons, all to demonstrate that we really don't see any UBE3A binding anywhere to chromatin. Again, it's negative data, so maybe we ultimately did all of this wrong. Our control chip said we didn't, but that's a caveat. So what types of genes are differentially expressed? And you know, there's 855 genes, and so what do you do with that information? And it is kind of hard. So ultimately, all we can do is ask what types of genes are upregulated and what types of genes are downregulated. And ultimately, the genes that we see downregulated are those involved in synapse development and whatnot, which largely suggests that the neurons are developmentally delayed, which is consistent with the electrophysiology. But the other thing we see is that the one insight into biology that we've learned from this is that there seem to be genes involved in extracellular matrix and signal peptides that are upregulated, and I'm intrigued by some of the results that Ben Philpott's lab has seen perineuronal nets, which really are involving extracellular matrix, and so maybe this is pointing a little bit at the biology in terms of a phenotype. So where I'm at now is I have mixed feelings about using the transcriptome as a phenotype, because on one hand, if a single gene disorder yields 800, more than 800 differentially expressed genes, I can only imagine what's happening when you're duplicating 20 or so genes in multiple copies. So the other thing that we learned from this, though, is that we got 800 and some odd genes when we use isogenic cells, so genetically identical except for one base pair change. And this told us that trying to do an RNA-seq analysis in non-genetically related typical and Dup15q cells is a really bad idea, because we can't know whether the gene expression changes are due to the background genetics or due to the duplication itself. So my lab has spent a crazy amount of time with very little success trying to make these isogenic cell lines. And this ends up being really, really hard. So the first strategy that we've been, I won't tell you about our first failure, but the second first failure, it involves interstitial duplications. We thought that this one might be more tractable, because what we decided to do here was to insert LOXP sites. LOXP sites are these great recombination sites that are used in mouse to make conditional deletions. And so you can use Cre recombinase, and it mediates a deletion involving these 2 LOXP sites. And so, you seat the LOXP sites. We thought we were being clever because it's the same sequence duplicated twice, and so you can use one CRISPR to make the cut. You can use the same targeting arms. We even, tricked this out with different positive and negative selection markers to help us do this more simply. And then once you get these LOXP sites seeded, you can use Cre recombinase and then a negative selection using ganciclovir, and you end up with a single quiet LOXP site, which is 37 base pairs inserted somewhere benign, and you end up with this isogenic control for your interstitial duplication. And we also thought, we'll take advantage of the fact that these are different alleles. The paternal allele came from a dad, and the maternal allele came from a mom, so maybe there's a heterozygous SNP. And we can use Cas9, a CRISPR that only cuts the maternal alleles. And sure enough, we found a heterozygous SNP in this cell line. This is the genomic DNA that shows that there are 2 A's and 1 G, and so if 2 G's and 1 A, and so if we can cut the 2 G's and leave the 1 A intact, it would be great. And so we designed, I am wrong enough to know that I like backup plans and backup plans to backup plans, and so we picked a site where we had 3 different CRISPRs that would work. 2 of them are dependent on this SNP, and 1 of them is not dependent on the SNP. And so thus far, we've only tried the ones that are dependent on the SNP. And like I said, I'm so good at being wrong that of several clones that we had with the 2 LOXP sites, none of them survived negative selection, which was a good indication that we were never hitting both LOXP sites on the maternal alleles. And so if I could diagram this, we were hoping for this scenario where you had the 2 Gs on the maternal alleles, and when LOXP recombined these out, you would end up with an A and a G. And instead, this told us that the situation is actually this, where you have a G on the paternal allele and you have G on one of the 2 maternal alleles and an A on the other maternal allele. And so while it was a failure, it told us something about these interstitial duplications, is that they're happening in meiosis 1, and you have 2 different maternal alleles. And I suppose it's something that the community had already, the geneticists at least, have already known. But nonetheless, this is going to be a confounder in a lot of what we do. So we can't rely on SNPs because the SNPs, the different maternal alleles, can come from different copies of mom's chromosome 15. So like I said, nonetheless, we have we still have one CRISPR that is not dependent on the SNP, and we fortuitously, because we study Prader-Willi syndrome, have found other CRISPRs that preferentially cut the maternal allele. We found these accidentally in trying to make SNORD116 deletions, and so we're trying the same approach with a different set of CRISPRs. So what about the IDEX? This is a little bit harder. So that strategy won't work with the IDEX because you actually have 4 copies of the genetic region. So that's a little bit too much to ask. But 2 papers came out over the past couple of years talking about CRISPR-Cas9 mediated targeted chromosome elimination. And so we wondered whether we could evict the IDEX chromosome. And in full disclosure, my first failure was a first failure to evict the IDEX chromosome. It doesn't leave on its own. We knew that. And so we asked, can we make it leave? And so our strategy to do this has been, we've designed CRISPRs that cut the Golgi 8 repeats. And the reason we thought about this is, so those other 2 strategies that were published, these guys each used CRISPRs that cut chromosome-specific repeats, and eventually those chromosomes left. And so in our case, we cut what we thought to be chromosome-specific repeats. So the Golgi 8 repeats are present in each of the breakpoints. They're present multiple times, and so the CRISPR is cutting a lot. And then we also added CRISPRs to cut SNORD116 and SNORD115 because those are also repetitive. Our thought is that, well, these are present in 2 copies on the IDEX chromosome compared to 1 copy on each of the parental chromosomes. And so maybe we're more likely to get the IDEX chromosome, or worst case scenario, it's just a numbers game. So we've nucleoffected 3 CRISPRs into IDEX 15 iPS cells. We select for the presence of the CRISPRs. And then we screen by looking at the copy number of UBE3A that's left. So if we've successfully lost the IDEX chromosome, we should only have 2 copies of UBE3A instead of 4 copies of UBE3A. We always run controls where we have controls that have 1 copy, 2 copy, and 3 copies, as well as 4 copies of UBE3A. And ultimately, we still haven't this one's really preliminary. Out of 20 clones, we didn't get one that has only 2 copies. But we got several that appear to have 3 copies. And so this is encouraging. So now we're asking whether, and what we think in these cases where they have 3 copies is that they've lost one of the 2 parental chromosomes. And while that's not great as a cell culture model, it is great as in proof of principle that we can get rid of a chromosome, and hopefully we can figure out whether we can lose both the maternal and paternal chromosomes to make sure we're not, it would be bad if we can only lose the paternal chromosome. And we do have a little bit of preliminary data that says we can lose a paternal chromosome. We don't have data yet that suggests that we can lose a maternal chromosome. So. But nonetheless, This is new, again, we've only screened 20 clones so far, but I think it's promising, and so we'll plot on. So if you'll indulge me for the last couple of slides here, I'd like to tell you about some of the work we've done in the epigenetic regulation of maternal and paternal 15q. Because again, in our studies of Angelman and Prader-Willi syndrome, we've learned some kind of interesting things about this chromosomal locus. So in studying, one of the ideas we had was to map the chromatin states of maternal and paternal 15q11 to q13 independently. We have these great cell lines that have deletions of mom's copy and deletion of dad's copy. And so what those let us do is look at, in the case of Angelman iPS cells, they have only dad's copy, and Prader-Willi cells only have mom's copy. And we performed a bunch of different ChIP-seqs, and I'm showing you the results from H3K4 trimethylation, which marks actively used promoters. So everywhere you see one of these brown, yellow peaks or whatever, that's where an active gene is starting. That's its promoter. And then we also did H3K9 trimethylation, which is a known marker of repressed genes. And so, and these are in iPS cells. And what we noticed is in our Prader-Willi cells with only a maternal allele, the H3K9 forms this really large block of repressed chromatin. So, and I should have backed up and said that these are histone modifications. These are methylation that happens on histone tails. So that's not DNA methylation, it's histone methylation. We see this large repressed block that spans the entire imprinted domain all the way, and it stops just short of UBE3A. And it's highly H3K9 trimethylated in Prader-Willi cells, but not in Angelman cells. And conversely, we see genes that are active on the paternal allele in Angelman cells that aren't active in the Prader-Willi cells. And so the two alleles have different chromatin. What's interesting, one of the other interesting observations we've seen is that this may affect neighboring chromatin. So this section here, where CIFIP, NIPA1 and NIPA2 are, they actually are present in both copies, but whether you have a paternal deletion or a maternal deletion is changing how the appearance of this chromosome, of this chromatin, and so that might be interesting going forward. But the real reason we do this is because in our studies of Prader-Willi syndrome, we've figured out that chromatin can be changed by manipulating chromatin regulators. So we've knocked out a gene called ZNF274 that brings in H3K9 trimethylation in Prader-Willi cells. And when we do this, we can change this whole block of chromatin. It's not exactly like Angelman, but it's more like Angelman, somewhere in between Prader-Willi and Angelman. And when we first did this, we noted that in the iPS cells, this doesn't lead to activated expression. If you look at the H3K4 trimethylation, there's only one gene, this happens to be MAGEL2, that's turned on now in the Prader-Willi cells compared to the ones without the chromatin modulator deletion. We only see one gene activated, but when we differentiate them into neurons, you see better activation, and this is specifically SNORD116, which eventually becomes the antisense transcript to UBE3A. So one of the thoughts we've been having is in DUP15Q, can we think of chromatin regulators to also turn a maternal allele into something that looks more like a paternal allele, and would that provide a more global regulation of gene expression in these cases? And so we think it's useful to understand the chromatin of the maternal and paternal alleles. And this has kind of led us to think about the fact that the maternal and paternal alleles look different in a nucleus, and they're also probably partitioned in different places in a nucleus. And so we've also thought a little bit about three-dimensional chromatin structure. And as part of a paper that we had published recently in PNAS, we did a technique called 4C-seq, and what this does is it allows you to pick a point and ask, what are the other places of chromatin that are interacting with that one spot, that one anchor spot in three-dimensional space, and in an unbiased way. So we picked this space upstream of UBE3A because there happens to be CTCF site, a chromatin organization site upstream of this. And if you look at the chromatin state of the space upstream of UBE3A, it looks like a lot of enhancers. And so this is kind of an extended enhancer activity space. And we wondered whether this is an enhancer for UBE3A. And this data doesn't necessarily tell us whether it is or it isn't, but one of the interesting things that we saw in this data was when we compared iPS cells to neurons. In neurons, this upstream enhancer state for UBE3A is interacting with the promoters for GABA beta 3 and GABA alpha 5. And so what may be the case is that they may share regulatory elements, and again, namely these enhancers that are here. Maybe they're enhancing both UBE3A and the GABA alpha 5. And so one of the things we can do with CRISPR is go in and change, activate these enhancer states or inactivate them directly, and we want to ask whether it will regulate both UBE3A and the GABA beta cluster to see whether or not, again, we can approach epigenetic modulators in this way. So that's the status of what my lab has been up to lately. And with that, I'll acknowledge the people who did the work and the funding sources. I have two excellent collaborators at UConn Health, in Eric Levine and Les Lowe, and they make it fun to be a geneticist because I can make a change and then they can do cool functional stuff with it and figure out like what our change did. And I wouldn't be able to do half of what we do without them. We work with the stem cell core and the human genome editing core that have allowed us to make all the CRISPR edits and whatnot that we do. I have to thank the families who've donated samples because we have iPS cells that allow us to study this tough genetic disorder without making them all ourselves. And then funding sources, the Angelman Syndrome Foundation, the NIH funds both Angelman and Duke15q work for us. The state stem cell Research Fund and Levo Therapeutics have funded the work I talked about today. And of course, our talented students who are the nuts and bolts of everything we do. Thank you.